Hello, everyone. My name is Christopher Leptak, and I'm the director of the CEDAR Biomarker Qualification Program at the FDA. I appreciate the opportunity to share with you some thoughts today around how biomarkers are used in drug programs from a regulatory perspective. The disclaimers is with any federal employee, we're not able to have any type of potential conflict of interest, whether that be financial or otherwise. What I'm going to do is start off with just setting a landscape, making sure that we all have the same understanding of the terms and what I'm going to be referring and taking us through what's called the BEST resource. Then we're going to talk more generally around how biomarkers are integrated into drug programs, and then specifically talk on one of those pathways, specifically qualification. I want to then bring to your attention some biomarker-related resources, and then we'll summarize at the end. So from a regulatory perspective, biomarkers are very clearly defined as a defined characteristic that's measured as an indicator of a normal or pathologic biologic process or response to an intervention. We think of biomarkers very broadly at the FDA. Many times people will think of something that can be measured in, say, a body fluid like serum or CSF or urine. We have a much broader approach, and that can also be radiographic biomarkers where you're looking at anatomic structures. They could be physiologic in nature where you're looking at the function of an organ system. So there's many opportunities. As long as it's a defined characteristic, it can be considered to be a biomarker. But it's not, though, is how a patient feels, functions, and survives. And that puts us into what we call the clinical assessment world, and specifically that contrasts it from what are called clinical outcome assessments, COAs. So the definitions aside, certainly there are many different communities that use biomarkers. Regulatory is just one of them. Also, they are used in clinical decision-making as part of patient care. So the clinical community is certainly very much on board with the use of biomarkers. And then there's the scientific and translational communities, which share their understanding of biology through publications in scientific journals. Many times folks will ask us, well, if one community can use a biomarker for a particular purpose, why can't all of them? And so I do want to bring to your attention kind of what makes our community different, meaning us as regulators. And it's not that one community is right or wrong. It depends on what the objective of that community is and what they're trying to achieve. And so from a regulatory perspective, we approve drugs from populations. And so we don't have the benefit of having individual conversations to use a biomarker to direct a patient's care, for instance. So as part of that, we need to make sure that there's adequate information and evidence to support a biomarker's use in order to have that information then be used as part of a drug approval that will be given to a population at large. The main things that we're looking at are reproducibility of the data. Certainly we all know that biomarkers are out in the scientific literature in great abundance, but very few of them actually rise to the level of rigor where they're used more broadly, either by clinicians or us as regulators. And that's not saying that the publications are wrong. It just means that the intent of a publication is different than, say, using it for a patient care. Likewise, when a biomarker is in play, it's measured in some way, and so we have to look at the adequacy of the analytic device around the biomarker's reliability. And then finally, from a sort of pragmatic way, we have to look at the feasibility of the biomarker should that drug be approved. So for instance, you're not going to want to give a common disease which impacts hundreds of thousands or millions of patients and have a biomarker measurement technology be a PET scan. Probably not the most pragmatic approach. So we do need to consider that as part of our regulatory use. The definition of a biomarker aside, there are other related terms that are certainly of great interest and are used as part of discussions of biomarkers. There was a joint collaboration between the FDA and the NIH a number of years ago, and ultimately they created what's called the BEST resource, Biomarkers, Endpoints, and Other Tools. It's on the National Library of Medicine. It is a living document that's periodically updated, and I would highly encourage you to look at that, and you can even provide feedback to the website. So ultimately, when it came to the biomarker subtypes or classes, it all came down to whether you are a lumper or a splitter, but ultimately what we did do was focus on classes that were differentiated based on how they were used. And so we ultimately came up with 7 different classes, and they're listed here. I think the best way to think about these classes is in a sort of a picture diagram that I call the pathogenesis of a biomarker, basically, and where you're trying to do a fit-for-purpose analysis where you're matching the class with what it's intended to address. So if you start in the upper left, the normal physiology, ultimately normal doesn't mean unchanging. And so even for an individual, over the course of the day or over a course of a period of time, a given biomarker may have natural variability associated with it. And understanding that variability is very much important. Likewise, there may be differences between individuals based on either genetic or environmental background. And so this normal piece is so important because ultimately if you're going to use a biomarker as part of a clinical trial and you're looking for changes in that biomarker, you need to know, are those changes normal variability or are they important? The only way to know that is to know what that normal variability is. In normal physiology, you don't have yet clinical disease. So our first class of best biomarkers is here, the susceptibility risk class. And ultimately, these are trying to tell you what is the likelihood that an individual will develop clinical disease at some point in the future. Ultimately, we as organic beings, we change over the course of time, and that's true at the cellular and molecular level. Those changes sometimes can result in altered physiology, and then ultimately that can result in phenotypic disease, being a constellation of recognized signs and symptoms. Once you have a disease, there are 3 different classes of biomarkers that can be important: diagnostic, monitoring, and prognostic. Diagnostic biomarkers can be used from 2 points of view. One is the capital D diagnostic, as I call it, where you're actually diagnosing the disease. And the other is the little d diagnostic, where you're diagnosing a clinical component or an anatomic component of a disease. So many times diagnostic biomarkers are used as part of the inclusion-exclusion criteria in clinical trials so that you're identifying the appropriate patients to enroll. Once a person has disease, especially if it's not an acute condition, it's self-limited, but it's more chronic in nature, that condition can then wax and wane where you have periods of exacerbation or quiescence. And so that's when some of the monitoring biomarkers can come into play. And then finally, for those conditions that are more long-lasting, we want to know what their clinical trajectory is. Like, what is the likelihood that a given person with disease will develop a clinical event in the future, whether that might be a transplant of an organ or a change in therapy. And so ultimately, that's when you get into the prognostic class, where you're trying to figure out what is the likelihood that that individual is going to have a certain event in the future that's of interest. Once the therapy is in play, we get into the remaining best classes, where ultimately what you're trying to do is take this clinical disease and either stop its progression or potentially even reduce it back towards not having the symptoms in the first place, or reversal of the disease. So once a therapy is in play, we can look at pharmacodynamic biomarkers, where basically you're trying to say, okay, with this particular therapy, is there a biologic change that we anticipate will be occurring? So say it's an antibody that's binding to a cell surface receptor. So that can get us into a pharmacodynamic type of context of use. The pharmacodynamic biomarkers do not, in and of themselves, guarantee that biologic change that you're observing is either beneficial or not. It just says it's a change. That in and of itself can be good for proof of concept, but I do want to make that distinction. Likewise, predictive biomarkers can help to stratify patients into those that are more likely to respond to a medication, or those that are more likely to have an adverse outcome. And then finally, we know that all medications are going to have unanticipated or unexpected off-target effects. That's when we get into the safety biomarkers, where they can cause organ toxicity. Once there's a potential for non-reversal or non-progression of the disease itself, we now get into the subset of response biomarkers that can ultimately become endpoints, potentially. And then if there's, in fact, improved clinical benefit, the standard by which we approve a drug, you then get a much smaller subset of response biomarkers referred to as surrogates or surrogate endpoints. For a biomarker program to be successful, there are multiple components. There's the biomarker itself, there's how it's measured, making sure that you have the appropriate patient population, and then all of your clinical design elements. I put this slide up because it just helps to emphasize that the use of a biomarker in a clinical trial is very much dependent on a lot of assumptions. Even our understanding of science has embedded assumptions in it. And it's helpful to kind of recognize that up front, so that if you're, not going to be successful, you can still learn from your negative results. And the only way to do that is to clearly define what your assumptions are going into the particular clinical trial. If you try to do it after the fact, it gets much messier. When we use a biomarker in a regulatory setting, we refer to what's called a context of use. Broadly and simply speaking, it's the best biomarker category, and then what is the biomarker's impact on the clinical trial design. Some of the examples are included here below. Many times the biomarker can be used as part of the inclusion-exclusion criteria, where you're trying to do some type of an enrichment strategy. It could also be used to alter treatment arm status, like if it's going to be predictive in nature. It could be for a proof of concept, for changing dose, and then ultimately endpoint or a surrogate. So these are just a few of the examples of different types of context of use that could be used. At the bottom of the slide is a description of the context of use for a recently qualified biomarker for total kidney volume. What you can clearly see is it really defines the parameters in which that biomarker can be used. And so that's helpful because once a biomarker is qualified, it can be used by then other drug developers for that same context of use without having to bring forward additional data for analysis. Some of the enablers for biomarker development include data standards. Certainly, we want to make sure that we have the same definition of disease, the same type of readouts, including the units for the biomarker, if those are appropriate, and so forth. The advantage of that is then that allows us to look at biomarker information from multiple sources and do comparisons across and potentially even pool them for some type of meta-analysis. We look at the data quality, of course, reproducibility. Data sharing can be exceedingly helpful since it's rare that a single data source has sufficient data to fully support the biomarker's use for a regulatory purpose. We look at the analytics. To the best that you can, try to standardize as much of the pre-analytic variables as you can through SOPs and storage and transport protocols. And then finally, we look at how the biomarker impacts the clinical trial elements themselves. Biomarkers can be brought forward in different ways for us as regulators. One is through the drug approval process, where a particular drug company will bring it forward under IND, and ultimately become an NDA or BLA. There's the scientific community consensus, which is ultimately the publications that are out in the scientific literature around the biomarker's use. It may also be rolled into as part of professional consensus statements. And then there's also the ability to qualify a biomarker in a way that's independent of a specific drug program that can then be used more generally across different programs and diseases. It's important to note that all these pathways do not necessarily exist in isolation. Many times a biomarker is employed in multiple of these pathways simultaneously. That's a true benefit for us as regulators because we get access to more information to make sure that everything seems to be heading in the right direction and under the same agreement. The information for these type of systems are all the same. We use the same definitions. Internally, they're all very much multidisciplinary teams that evaluate the data. So there is consistency across these. I will say that scientific community consensus is usually where we start for hypothesis testing, because it's basically saying, what do we know about this disease or the pathogenesis or the mechanism of action of a therapy? That then drives how a biomarker might be used in a drug program specifically or brought forward as part of a biomarker qualification effort. So I mentioned that because it does talk about the interrelatedness of these multiple pathways. The value proposition for biomarkers is that ultimately, if it gets qualified, it can be used for its context of use without the need to reproduce or resubmit data. And likewise for us, we don't need to reevaluate data for that context of use. Since it is independent from a drug-specific program, there's an opportunity for a collaborative effort to bring information forward, and that can include academics, patient advocates, non-drug industries, government organizations. So there's an opportunity for a sort of pre-competitive collaborative environment to work together to tackle the potential problem. There's also the opportunity for data sharing and expertise that can be extremely beneficial. And then ultimately, qualification can advance the scientific understanding in a non-competitive way, so that hopefully multiple stakeholders will be engaged, willing to contribute, whether it be data or resources, to help increase the scientific understanding for the biomarker to be used for a regulatory purpose. And then ultimately, DDTs, depending on their context of use, can certainly allow for the completion of clinical trials at lower cost or with fewer patients. When you're looking at developing a biomarker for regulatory purpose, it's important to think about sort of what are the components of that development effort. The first is the need statement. Here you're clearly articulating what is the current paradigm and that could be improved upon, and how would this potential biomarker benefit that. And you really have to clearly articulate that need statement, because here's where you're trying to convince us as regulators that it's a good investment of our resources to help work with you through this development effort. That need statement has to be focused on a drug development use need, not just needs in general. So for instance, if there's a need to help physicians better understand how they're going to treat an individual patient, that's not related to drug development specifically, and that probably wouldn't be the best need statement to be brought forward. Next, we get to the context of use, which we covered on the previous slide. And then ultimately, based on the need and the context of use, we then try to figure out what evidence we need. And as part of that evidence, we do look at the benefits and risks to patients, either in the trial or the may ultimately receive the drug that's approved. And then for the evidence, we want to definitely look at the analytical and clinical validation that I'll speak to in a moment. We're going to look at information as it exists, and then also try to figure out what information we need to support that particular use in drug development. For the analytics, we broadly break it down into two halves, the analytical and clinical validation. The analytical simply says that you can reliably measure something. And then the clinical validation says that that something that you're measuring actually has biologic or clinical meaningfulness. That's simplifying it down a lot, but it is a nice kind of simple way to make the distinction between the two. As part of a biomarker development effort, we spend a lot of time on the analytical validation early on, because if you can't reliably and reproducibly measure something, then you can't really see much about clinical validation until those parameters have been explored. So the analytical measurements of interest, they're listed here. It's not a complete or exhaustive list, but it does bring forward a number of the things that are important. Depending on the context of use and also the potential risk for patients in the trial or they don't receive the therapy, that kind of drives the level of analytical validation that needs to be conducted. And when it comes to the type of device, some of them may be FDA cleared or approved. Some of them may be more in a laboratory developed environment. And then there's some that are for research use only. You know that even if you're using an FDA cleared or approved method, you have to make sure that the way you're using the device in your biomarker effort matches the indication for which the device was originally cleared or approved. If there's a change, then we may need to see some additional data because you're using it in a way that it wasn't originally evaluated for. For biomarker qualification, we follow what we call the 507 process that was put forward in the 21st Century Cures legislation that was passed by Congress back in December of 2016. The Cures legislation, as well as the PDUFA negotiations at that time, put FDA front and center as an active participant in a collaborative way to develop these drug development tools, or DDTs as they're called. It also formalized a three-step submission process: the letter of intent, the qualification plan, and the full qualification package. More on those in a minute. It's a transparent process, which ultimately information about the submissions that we receive ultimately go up on our website, and the link is provided here. along with biomarkers when they're qualified, are also made public through the website. In addition to information about the submissions, we also post the recommendations that we provide to that particular biomarker developer so that they know what they would what they would need to do to be able to get into their next submission stage. Because all of this is available on our website through the transparency provisions, what we found is that it's really resulted in a shared learning culture so that ultimately everyone can learn from each other, and it's really increased the quality of our submissions within a relatively short period of time. So that, for instance, if you're developing a imaging biomarker using an MRI technology, you could look at other MRI technology biomarkers and look at what recommendations we've sent and incorporate those recommendations into your own particular development effort, thereby saving your time and energy. The 3 submission types have target review clocks internally, and we had to do that as part of Secure's legislation. So for letter of intent, it's 3 months, qualification plan is 6, and a full qualification package is 10. Certainly, if we can turn these around faster, we certainly do. And many times we are able to do that, albeit even in the current environment under COVID, obviously resources are sort of being redirected. So we're trying to stick to these timelines, but there may be some extenuating circumstances. The acceptance of the biomarker is ultimately based on its scientific merit. And that does the proposal address an impactful drug development need, much like what we talked before? Is there enough information to suggest there's a likelihood of success? Then what is the feasibility? Should the biomarker ultimately be approved? We do prioritize our submissions based on the severity and rarity and prevalence of the disease, much like what we would do in the drug realm for accelerated approval. And then it is possible that we can also triage based on public health priority, much like what we're doing now in the setting of COVID. In terms of the 3 submission types, the letter of intent was mostly focused on a clear description of the drug development need, the context of use that addresses that need, and then information about the proposed biomarker and summary level information about the measurement method that's available. It's a relatively short document. It's maybe 10, 20 pages tops. It's not meant to be several hundred pages and basically a cut and paste from an extramural grant to the NIH. This is a really tight, focused proposal. And information about that submission is presented in summary form. This is not a data analysis or primary data submission. Once you have an accepted letter of intent, you can then go on to the qualification plan stage, or the QP. Here's the qualification plan is really the soup to nuts of what we understand of the science as it exists today. What is our ultimate goal based on the context of use? What are the scientific gaps to get us to that goal? And then how are we going to address those gaps? And so transitioning from a letter of intent to a QP, we understand is kind of our biggest hurdle, if you will, because this is when the rubber hits the road. And so there is an opportunity for a lot of collaborative engagement as you transition from an LOI to a QP, and I'll mention those in a moment. It is important also that when you're developing your QP, that you really address the comments and recommendations that we put in our LOI determination letter. We take a lot of pride in putting together thoughtful comments to help the outside party really use their resources judiciously. And the best way to do that, sort of follow our recommendations, and if you're not clear what those recommendations are, there's an opportunity to have a conversation with us around them. And then finally, the FQP, or the full qualification package, is submitted. That has all the data, your interpretation of the data, and it's submitted to us in earnest. And then we look at it to see if we can, one, recreate the analyses that you've done, and see if we agree with the determinations that you make from those analyses. Internally, qualification submissions undergo a 3-step review. The first is that there's a program assessment looking for administrative completeness, and also making sure that you've sort of followed the content outline elements that are on the website. Once a submission is reviewable, the review clock starts, and then we bring in our subject matter experts, whether that be from the clinical divisions, from pharmacology, toxicology, clinical pharmacology, statisticians, analytics experts, whoever would have expertise that would be important for that particular submission. We're also going to look at regulatory precedents to see if this particular submission or this particular biomarker has been used in other contexts, how this proposal is similar or different. And then ultimately, the program and the subject matter experts put forward a recommendation of whether or not to accept a given submission. And then regardless of whether or not that submission is accepted or not, we do provide pretty extensive recommendations of how that particular biomarker can be developed further. The decisional body is the CEDAR DDT committee, which is made up of senior leadership from our center or their representatives. And it gives an opportunity for broad senior input early and on in the process. And it helps to increase consistency across therapeutic areas and divisions. In terms of engaging with us, there's an opportunity to have either a pre-LOI or QP meeting. We use these meetings to try to help folks better understand either the process or what's expected in a given submission type. And we can also tailor these to specific discipline questions, whether they be stats or analytics, et cetera. We can have post meetings, and once we make a decision on a given submission and you've received your determination letter, we can end up having a debrief meeting to go over the written recommendations to make sure that we can clarify any of the comments that we made. And then finally, there's an opportunity to have a pre-FQP meeting where you're looking at any challenges or updates that may have occurred since you had your qualification plan accepted. The program itself, we do a lot of coordination between our centers. Initially, biomarker qualification was a CEDAR-driven process. With the advent of cures, it's now a two-center process, so CEDAR and CBER. And so the context of use can be appropriate for sometimes both of our centers or maybe just one or the other. Because other other regulatory bodies do have qualification programs, namely the EMA. We do have a monthly meeting with them where we talk about program process and project issues, especially if there's a parallel engagement between both regulatory bodies. The content outlines for the 3 submission types are available on our website, and was the final guidance that was put forward around the process and describing it. And there are a couple of guidances that are currently in development around the evidentiary framework, the analytics and surrogate endpoints, and then there's also a CEDAR BQP map internally. So with that, I very much thank the organizers for the opportunity to present to you today, and I wish you all well.